Hello and welcome everyone. In this podcast we're going to talk about the structure of DNA as well as the structure of the smaller components of the DNA molecule, the nucleotide. So let's begin by drawing a double-stranded piece of DNA here. Of course this is DNA. Now to fully understand this structure of DNA, we need to understand the smallest component of DNA. And then that will help reveal what the entire macromolecule of DNA looks like and the rules that govern its shape. So the first thing we're going to want to do is we're going to want to talk about the nucleotide. And as a quick definition, this is the repeating structural subunit of DNA or RNA. So we want to talk about that first. Then we're going to want to talk about how these nucleotides, and sometimes I'll abbreviate nucleotide just as an NT. So we want to understand how these nucleotides bind to each other to form a single strand of nucleic acids. And sometimes I'll abbreviate nucleic acid with just an N and an A. The next we'll want to ask is, how do we take single strands of DNA and create the double helix? And as we're going through this, we'll point out the differences between DNA and RNA. So let's begin with talking about the nucleotide. So the nucleotide is made up of three structural parts. The first is a phosphate group. The next is a sugar. And the last is a base. Now if this nucleotide is going to make RNA or DNA, the phosphate's always going to remain the same. It doesn't change between the two different types of nucleic acids. However, the sugar does change. So let's talk about the two different sugars that we would find in DNA or RNA. So let's start looking at the sugars. And there can be one of two kinds of sugars, depending on if you're talking about DNA or RNA. For DNA, the sugar is called deoxyribose. Let's go ahead and draw that here. It's a ring structure. And just, if you haven't had organic chemistry yet, just so you know what we're looking at here is we always number the carbons. And in these ring structures, as we draw them, there's always a carbon here at that point, this one, and this one. And we should draw this one up here. There's another carbon here, not in the ring. We number these. We say this is the primary carbon, this is the secondary carbon, this is the tertiary, quaternary, and then this is the 5' carbon. On these carbons at these points, there will be hydroxyl groups added to them. Except for this one in DNA, it is just hydrogen. We have a hydroxyl group here too, and then we have one here that I've already drawn. Next, let's draw the sugar that we find in RNA. RNA sugar is called ribose. And so let's draw that here. And you'll see it's very similar to the deoxyribose. We have our hydroxyl group here off of our carbon 1. The difference comes here at carbon 2. At carbon 2, instead of a hydrogen, we have another hydroxyl group. Then off of this carbon 3, we have a hydroxyl group, and this is carbon 4, carbon 5 with a hydroxyl group off of it. So really the only difference between deoxyribose and ribose is the hydroxyl group at the 2' in ribose and the lack of a hydroxyl group at the 2' position of deoxyribose. It may seem like a very simple difference, but it makes a huge difference upon how RNA behaves and how it functions. This hydroxyl group here makes RNA much more sensitive to certain enzymes known as RNases. And we'll talk about these later in the semester. These RNases degrade RNA, and the reason they degrade RNA is because they recognize this 2' hydroxyl group. And it doesn't recognize DNA because it doesn't have that 2' hydroxyl group on the deoxyribose. Okay, we've talked about the phosphate group. We said it doesn't change between DNA or RNA. And we just talked about the sugars and talked about how that's different between DNA and RNA. Next, let's talk about the bases. And in doing so, I'm going to erase our sugars here. And the bases can either be pyrimidines or purines. The pyrimidines can be one of 3: cytosine, and we often just abbreviate that with a C; uracil, which we often abbreviate with just a U; and thymine, which we abbreviate as a T. Purines can be one of 2. They can be guanine, which we often abbreviate just as G, or they can be adenine, which we often abbreviate as just A. I always remember these because when I think of the word pyrimidine, I think of a pyramid. And I'm not a very good drawer, as you probably have already realized, but I think of a pyramid, like so. And if I think of a pyramid, I think about this point on the top. And I think of that being very sharp, and if you touched it, it could cut your finger. I know it's kind of corny the way I remember things like that, but that's always helped me remember that the pyrimidines are C-U-T, spelling out cut, because the top of a pyramid is sharp and could cut your finger. And then I just remember the purines are the other 2. No corny way to remember that. Another thing you will have to remember is that, I'm not going to make you, with the sugars, you should be able to recognize those structural differences. And you should be able to draw them as well. With the pyrimidines, you don't need to recognize their structural differences and you won't have to draw them. But you should remember that the purines are a double-ringed structure. And I'll draw one of the double rings like so. And you should remember that the pyrimidines are a single-ringed structure. You won't have to draw them, but if you see them present and I ask you, is this a pyrimidine or a purine, you'll want to ask yourself, does it have 2 rings or 1 ring? If it has 1 ring, you know it's going to be one of the pyrimidines. If it has 2 rings, you know it's going to be one of the purines. The way I've always remembered that the pyrimidines are a 1-ring structure and purines are 2-ring structures is that the word pyrimidine is a longer word, yet the structure is smaller than purine, which is a shorter word and its structure is larger. So it's kind of counterintuitive. One last thing you need to remember about these different bases is which nucleic acid are they found in. So in DNA, you will find the pyrimidines cytosine and thymine, and the purines guanine and adenine. In RNA, you'll find the pyrimidines cytosine and uracil, and the purines guanine and adenine. So the only difference in the nucleotides between DNA and RNA is that DNA has thymine and not uracil, and RNA has uracil and not thymine. Okay, so now let's take 2 nucleotides and bring them together in an effort to build a single strand of DNA. And so let's start by drawing a nucleotide. It's going to be one that has thymine on it. We have our phosphate group here at the 5' position. At the 3' position we have our hydroxyl group, and it's DNA, so we have our hydrogen here. Now let's draw the next nucleotide in just a different color, just to try to keep them separate. And we're going to draw it, for now anyhow, like so. We have our sugar here, our deoxyribose, because there's our hydrogen. And let's go ahead and make this one a guanine. Doesn't have to be, that's just what I chose. We're going to have our hydroxyl group here, and then we're going to have our 5' phosphate group here. And I also want to remind you that this is our 3', and over here is our 3'. That's important to remember as we go forward, and I'll explain why in a moment. As these two nucleotides come together, they will form what is called a phosphodiester bond. Or linkage, you could call it as well. Right? And this sets the stage for this single strand of DNA to form. Let's go ahead and add one more nucleotide to the bottom here. And we'll make this one cytosine. Again, it's DNA, so we have to have the hydrogen there. We have our 3' hydroxyl group. And then we have our 5' phosphate group here. Again, we're going to form a phosphodiester linkage here. And then, we don't have room here, but we'll keep adding new nucleotides to this growing strand in this direction. And a DNA strand is always written with 5' at the top and 3' at the bottom. And when we say 5' to 3', we say because at the top of it we have our 5' phosphate group, and at the bottom, regardless of how long this strand is, there will always be a 3' hydroxyl group. Now we know that DNA is a double helix, like so. Now this little double helix here, I'm going to draw it in kind of a ladder format, like so, because it'll illustrate the point I'm trying to make here. We know that one of the strands is 5' to 3', just like we drew over here, and we know the other strand is reversed. We know that this strand goes 5' to 3'. We call this being anti-parallel. So now as we begin to draw the other half of this double helix, we have to keep in mind that it's going to run reverse. We're going to have our 5' phosphate at the bottom and our 3' hydroxyl group at the top. So really, the strand is going to be reversed and kind of upside down. To help me draw this, I'm going to take my board and I'm going to turn it upside down. Okay, I'm going to begin by drawing a base, a nucleotide I should say, right across from here. And I'm going to begin by drawing a deoxyribose sugar, like so. And it's going to have a base here. I'm not going to tell you which one it is yet. It's going to have a hydroxyl group here, just like we've seen before. It's going to have this 3' hydroxyl group here. And up here we're going to have our 5' phosphate. It's going to attach to the next base, just like we saw on this strand here. Let's go and draw the sugar first. And again, there's going to be a base here. I'm not going to tell you what it is yet, but there's a base there. This phosphate group is up here at the 5' end of this nucleotide. And then we have our hydroxyl group here at the 3' carbon. Just like we saw over here, we're going to form this phosphodiester bond in between them. It's a covalent bond, so it's a very strong bond to attach them. Okay, let's draw another base here, or another nucleotide I should say. And it's going to form that phosphodiester bond between this 5' phosphate group and the 3' hydroxyl group. You'll notice that we have our 3' hydroxyl group at one end and our 5' phosphate group still up here. And it's opposite with the other strand. Again, as we said before, it's written upside down now, but it's anti-parallel. So now what's going to bring these two strands together? We can see how we've created two separate strands here, but what's going to hold them together? What's going to hold them together are going to be hydrogen bonds in between the two bases. And there was a scientist, his name was Chargaff. So let's write it right here. Chargaff. Famous French scientist, and he came up with a set of rules that we now call Chargaff's rules. And those rules tell us that cytosines always bind with guanines. And likewise, guanines are always going to bind with cytosines. Thymines are always going to bind with adenines. So as you'll notice, we always have a pyrimidine binding with a purine. Pyrimidine, purine, pyrimidine, purine. It needs to be that way. It needs to have one of the bases being a 2-ring structure and another base being a 1-ring structure. In doing so, you have this consistent space between the different strands. Another thing we should point out while we have this drawing up is that what's holding these together, let me go ahead and put boxes around these bases here just for consistency. What's holding these together are hydrogen bonds. And we know that there are 3 hydrogen bonds between guanines and cytosines. And we know between thymines and adenines, or the reverse, adenines and thymines, there are 2 hydrogen bonds holding them together. I would say you should be able to draw something similar to this. It won't be on the quiz. It could be part of the activity in class tomorrow, or it could be on your exam. But you should be comfortable drawing it at this level of detail. So don't worry about drawing the structures of the bases. G's, C's, A's, and T's are sufficient. But you should be able to draw the sugars. And it's really important to get used to thinking about the 5' and the 3'. These are very important. Every time we write a nucleotide, when we talk about replication or transcription, we're going to refer back to the 5' and 3' parts of these molecules. So it's a good idea to go and get that firmly established in your brain right now. And you should, if we're talking about DNA, you should always make sure you put a hydrogen here at the end. Okay, that ends this podcast. So let's summarize real quick what you learned from this podcast. You learned that nucleotides, NT for short, are made up of a phosphate group, a sugar, and a base. You learned that when we string these nucleotides together, that we form a single strand of DNA. And you learned that this bond holding these together is called the phosphodiester bond. You also learned that when we write nucleotides like this, that we always write them in a 5' to 3' direction. We also learned that these nucleotides that are linked to each other in these single strands form hydrogen bonds with another single strand to form the double helix. And you learned that when we write these, again we said 5' to 3', but the other strand is always written in an anti-parallel way. So 5' to 3' in one direction, the other direction 5' to 3'. We learned that the bases here in DNA are guanine or adenine, or cytosine and thymine. And then in RNA, they are also guanine and adenine, or cytosine and uracil. We also learned that in DNA that the sugar is deoxyribose, and in RNA the sugar is ribose. You also learned that guanines and adenines are purines, And cytosines, thymines, and uracils are pyrimidines. So I guess we can write those under here in small letters. You learned that cytosines with 3 hydrogen bonds will bind with guanines in a double helix, and that thymines will bind with adenines, or uracils will bind with adenines with 2 hydrogen bonds. One way I always remember which binds with which is that the T and the A, these 2 letters have straight lines, so they go together, whereas the G's and the C's have these curved lines, and so they bind together. Just a silly way I've learned to memorize this. That covers most of what you're going to need to know about the structure of nucleotides and the double helix. Alright, well, until the next podcast, I'll talk with you later, and if you have any questions, please let us know.